Hello friends, welcome to ENTRANCEADVISER.com. Today's topic is molecular basis of inheritance, that is the part of chapter 6 and the lecture 2. And in this unit, I'll be discussing about mainly the replication and transcription in details. So let's start. If you are new to this channel, please hit the bell icon and do subscribe to our channel for more videos like this. Let's start the video. Cells divide during certain phase of the cell cycle. Before the cell cycle, doubling of the DNA occurs to produce two daughter DNA molecules in the synthetic S phase of the cell cycle. These daughter DNA molecules are identical to each other and these are also identical to the parent molecule. As the genetic informations are contained in the DNA, the stability of the DNA molecule during its division is very important, and so that the biological informations are truly represented in the daughter molecules. This special property of the DNA is known as autocatalytic activity, which is shown by the DNA molecule as the genetic material. DNA is always present in the double helical structure except the bacteriophage into that is the lambda 174, where the DNA is single helical. Okay. The two strands of DNA run antiparallel. As you can see here, the two strands of DNA run antiparallel. It's 3' and here 5'. Here it is 5', here it is 3'. So the polarity, that is their 3' and the 5' polarity are opposite, and these DNA strands exist in complementary base pairing between one purine and one pyrimidine base. These properties of the DNA are unique and are very important to establish its self-replicating attribute. So, the DNA replication is the process by which the DNA makes a copy of itself during the cell division. Watson and Crick suggested that two DNA strands of DNA separate from each other and act as a template for the synthesis of the new complementary strands. After the completion of the replication, each DNA molecule would have one parental and one newly synthesized strand. This method is called semi-conservative mode of replication, the process by means of which the DNA molecule divides into two identical daughter cells, daughter DNA molecules as well, using the two strands of the parental DNA as the templates following which the deoxyribonucleotides make. The complementary base pairing is known as the replication. Now I will explain about the general features of the DNA replication. The DNA replication occurs bidirectionally. The DNA replication initiates at a point known as the origin from which the DNA synthesis proceeds at both the directions. And the DNA replication occurs from 5' to 3' direction. As you can see, it occurs during 5' to 3' direction. And due to the presence of OH group at the 3' end only, here in the 3' end only, therefore the direction of the DNA synthesis is towards 5' to 3' end. And both the strands of the parental DNA acts as the templates. Both the strands of the parental DNA get separated and become single helical to be the templates, following which the new deoxyribonucleotides are incorporated by complementary base pairing. The unit of DNA replication is known as replicon. The unit of DNA in which the individual act of replication occurs is known as the replicon. Each replicon has one origin and two termini. In prokaryotes, there is only one replicon and DNA synthesis proceeds from a point of origin. But in eukaryotes, there are many replicons and many points of initiations. And the DNA replication occurs by a semi-conservative method that is very important. The parental DNA strands get separated by the destruction of hydrogen bonds between the nitrogen bases. Following the parental template DNA, new strand of DNA is synthesized by complementary base pairing so that the daughter molecule one DNA molecule of their and single strand remains as the parental strand and other strand remains as newly synthesized. Therefore, half of the parental strands is conserved in daughter DNA molecule and the DNA synthesis is semi-discontinuous. This is also very important. In one DNA strand, synthesis occurs continuously, but in other strand of DNA, synthesis occurs discontinuously. Therefore, considering both strands, the overall DNA synthesis is known as semi-discontinuous. And last but not the least, the DNA synthesis requires RNA priming before the DNA synthesis from a template short segment of RNA known as the RNA primer is synthesized. Let's move on to the next slide. Here is the proof, which is experimentally performed by Messelson and Stahl, shows experimental evidence of semi-conservative replication by growing E. coli, Escherichia coli on a nutrient media containing the nitrogen salt 15NH4Cl level with a 15N nitrogen. 15N was incorporated into both the strands of DNA, and such a DNA was heavier than the DNA obtained from the E. coli grown on a medium containing 14N. Then they transferred the E. coli cells to a medium containing the 14N. After one generation, when one bacterial cell has multiplied into two, they isolated the DNA and evaluated its density. Its density was intermediate between that of the heavier 15N DNA and the lighter on the 14N DNA. This is because during replication, the new DNA molecule with 15N old strand and a complementary 14N new strand was formed. Semi-conservative replication, so its density is intermediate between the two. Here we go. Replication of the DNA requires enzyme DNA polymerase that catalyze the polymerization in one strand of to 5' to 3' direction only after unwinding with the help of helicase enzyme. So the replication in one strand is continuous and other strand is discontinuous to synthesize the Okazaki fragments that are joined together by the enzyme DNA ligase. As I've told, the DNA replication is semi-discontinuous too. In this slide, I'll be explaining about the experiment schematically. The DNA hypothesis was DNA replicates semi-conservatively, and the method was number 1, grow the bacteria in nitrogen heavy medium. Then transfer some bacteria to this medium of 14N light medium, and bacterial growth occurs in both the medium, and after a generation, before the bacteria reproduce for the first time in the light medium at 0 minutes, the DNA parental is heavy, and take some samples after 20 minutes and 1 round of replication, and 40 minutes 2 rounds of replication. And they found the sample at 0 minutes parent heavy, sample after 20 minutes the first generation, that is all the intermediate are heavy, and sample after 40 minutes the second generation half intermediate half light. So the results are as follows. After 2 generations, half the DNA was intermediate and half was light only. There was no heavy DNA. As you can see here, it is not present. The heavy DNA was not present. One is intermediate, this is intermediate, and this two, this is 25%, these are 25, and this is 25, total 100%. Or 50-50, you can say, the parent strand and the new strand. The conclusion was that this pattern could only have been observed if each DNA molecule contains a template strand from the parental DNA. Thus, the DNA replication is semi-conservative. This is one, and the experiment is further explained in this slide also. If you refer to this, this is also good. The growth of bacteria in 15N and centrifuge, then transfer to 14N, the growth for 1 generation, then centrifuge DNA in CSL medium, and hybrid DNA was observed, as well as with the parental strand growth in the second medium after hybrid DNA. In the first generation, intermediate was observed, and in the second generation, 50-50. Now, I'll be explaining in more detail. The first step in the DNA replication is to unzip or unwind the double helix structure of the DNA molecule. This is carried out by an enzyme called the helicase, which breaks the hydrogen bonds holding the complementary bases of the DNA together, A with T, C with G. As you know, there are double bonds and there are triple bonds. And the separation of the two single strands of the DNA creates the Y shape called the replication fork, and the two separated strands will act as templates for making the new strands of the DNA. One of the strands is oriented in the 3' to 5' direction towards the replication fork. This is the leading strand, and the other strand is oriented in the 5' to 3' direction away from the replication fork, and this is the lagging strand. As a result of the different orientations, the two strands are replicated differently. This is an illustration to show the replication of the leading and lagging strands of the DNA, as you can see in the figure. So the synthesis of DNA in the prokaryotes and eukaryotes occurs essentially by similar mechanism. The difference is lying in the involvement of the enzyme in the two systems only, and the DNA replication starts at a single point known as the origin of replication. But in eukaryotes it starts at many initiation points of origin, and the common phenomenon of the replication is discussed here. And the DNA synthesis occurs in the 3 steps, mainly: initiation and chain elongation, and at last the termination. In initiation, the beginning of the DNA replication requires an uncoiling of the molecule, which is catalyzed by the gyrase enzyme, and formation of the single-stranded structure mediated by the helicase enzyme. You can say this is unwinding. Here it's unwinding taking place, and by the breaking of the hydrogen bonds in between them and between the end bases of the two strands. The two single helical DNA that formed act as template, which direct the synthesis of the nucleotides in the DNA in the new strands. And at the beginning, a small segment, 8 to 10 nucleotides long, the RNA is synthesized by the formation of the complementary base pairing of the ribonucleotides with the template strand. This segment of RNA is known as Rna Primer. It's very necessary. It's synthesized by the enzyme complex primosome, primase enzyme, which with some protein factors, and the Rna primer provides 3' OH, and which is later extended to the DNA, into the DNA. And the chain elongation, you can see the 3' OH group, here it is, of the Rna primer is extended by the incorporation of the deoxyribonucleotides, which are joined by the phosphodiester bonds and mediated by the DNA polymerase enzyme. The DNA synthesis starts at the midpoint of the replication unit or the replicon and proceeds towards 3' end of the growing chain. And the replication unit is looking like a bubble sometimes. Here you can see or eye, and half of it appears as a fork. In the fork, in one strand, synthesis of the DNA occurs towards the base of the fork in the direction of the opening of the double helix. In this strand, the DNA synthesis occurs continuously, you can see, and in this it is called the leading strand. And in the other, the strand, DNA synthesis occurs discontinuously. It's written here. And as a template strand is not available continuously, this results in the formation of the small fragments. These are the small fragments known as the Okazaki fragments, named after the discoverer Okazaki. This DNA strand is known as lagging strand, as it lags in the replication process. In the lagging strand, the DNA synthesis initiates at many steps, and for the synthesis of each Okazaki fragment, one Rna primer and one set of polymerase enzyme is very essential. On the contrary, for the synthesis of the leading strand, only one Rna primer is synthesized and only one set of polymerase enzyme is required. Later, the Rna primer is destroyed by the action of a special type of DNA polymerase enzyme, and the gap of the deoxyribonucleotides and the synthesized by catalyzing by this polymerase enzyme. Let's move on. Here in this slide, I'll be discussing about the difference between the leading strand and lagging strand. The leading strand, it is formed continuously, as a single fragment, and it requires only one primer to initiate the growth. That is, a short piece of RNA called a primer, produced by an enzyme called the primase, comes along and binds to the end of the leading strand. The primer acts as the starting point of the DNA synthesis. The DNA polymerase binds to the leading strand and then walks along it, adding new complementary nucleotide bases A, C, G, and T, and to the strand of DNA in the 5' to 3' direction. DNA ligase is not required here. This sort of replication is called continuous, as I have described earlier. In the lagging strand in the beginning, it is formed in the form of small fragments called the Okazaki fragments, and every fragment requires separate RNA primer to initiate. And numerous RNA primers are made by the primase enzyme and bind at various points along the lagging strand. Chunks of DNA called the Okazaki fragments are then added to the lagging strand, also in the 5' to the 3' direction. The DNA ligase required to join DNA fragments. This type of replication is called discontinuous, as the Okazaki fragments will need to be joined up later. Here is a picture provided. You can see this. So, at a glance, the parent DNA molecule consists of two polynucleotide chains held together by the bonds between the complementary and the nitrogen bases. An enzyme breaks the bond between the two polynucleotide chains of the parent molecule. Another enzyme pairs new complementary fuse with those in the two parental chains. Two DNA molecules form, each containing new chain, the green one. Okay. And in the termination, this happens when the growing forks of the two adjacent replicons meet at a point of the termination. The breakpoints between the Okazaki fragments are restored by the fusion of the broken ends with the action of the ligase enzyme, and thus the DNA synthesis is complete. In this DNA, one strand is parental and other strand is newly formed. So the mode is known as semi-conservative. The DNA synthesis is also known as semi-discontinuous, as the synthesis of DNA occurs continuously in the leading strand and discontinuously at the lagging strand. So this is very important for your exam, the difference between the lagging strand and the leading strand, or the leading strand between the lagging strand. Here are two differences given from a very authorized community. You please go through this. So once all the bases are matched up, A, T with C and G with called the exonuclease strips away from the primer, the gaps when the primer will and then filled yet more to be complementary nucleotides. It's a very good picture provided here. This is the DNA polymerase enzyme, and these are working along. There are helicase, there are lagging strands, this replication form, very detailed. And the new strand is proofread. It's to check or correct, to make sure there is no mistakes in the DNA sequence. Finally, what happens? An enzyme called the DNA ligase fills up the sequence of the DNA into two continuous double strands, and the result of the DNA replication is two DNA molecules consisting of one new and one old chain of the nucleotides. This is why the new DNA replication is described as semi-conservative. Half of the chain is a part of original DNA molecule, half is the brand new. And following replication, the new strand automatically winds up into a double helix. I hope you understood very clearly. I've explained into much detail and in multiple ways. Now coming to the mode of transcription. So you need to know the definition first. What is it? The process by means of which the information contained in the template strand of DNA is copied into a single-stranded RNA molecule of the complementary base sequence of the DNA template is known as transcription. An RNA strand that is produced from and is complementary in base sequence to a DNA template strand is known as transcript. General features of the transcription I am going to explain in a few moments. Before that, please refer to this description also. The DNA describes the transcription describes the process by means of which the genetic information contained within the DNA is rewritten into messenger RNA, mRNA, by the RNA polymerase. This mRNA then exits the nucleus, where it provides the basis for the translation of the DNA. By controlling the production of mRNA in the nucleus, the cell regulates the rate of gene expression, which is a very important part of the biology or the molecular biology. It is a process of copying genetic information from one strand of DNA to RNA. It's a part of the central dogma, as you can recall. It's DNA to RNA, simply this. And in transcription only, one segment of DNA and only one strand is copied into the RNA, not a whole, okay? The adenosine forms the base pair with the uracil instead of thymine. So the precursors of RNA synthesis are 4 ribonucleoside, 5 triphosphates, adenosine triphosphates, the ATP, the guanosine triphosphate, the GTP, and the cytidine triphosphate, CTP, and the uridine triphosphate, UTP. Only one strand of DNA is used as a template for the synthesis of the complementary RNA chain in the given region, as I've told you. The same strand of the DNA may not act as a template strand throughout the entire chromosome through all the stages of the life cycle. Keep it very clear in your mind. Print it in your mind. And RNA chains can be initiated de novo without any requirement of a pre-existing primer strand. The synthesis of the RNA chain occurs in the 5' direction with the addition of the ribonucleotides to the 3' OH group at the end of the chain. There is a reaction. This reaction is catalyzed by the RNA polymerase enzyme. I'm discussing about this. Okay, and the RNA polymerase binds to the DNA at the specific nucleotide sequences known as the promoters. If the transcript is an mRNA molecule, it is known as the coding strand of the RNA, as the triplet bases of the mRNA may code for the protein synthesis. The mRNA transcript is also known as the sense strand of the RNA, as this mRNA makes sense by specifying the sequence of amino acids in the polypeptide. An RNA molecule that is complementary to the mRNA is referred to as antisense mRNA. So what is RNA polymerase? The RNA polymerase is the main enzyme involved in the transcription. It uses the single strand DNA to synthesize a complementary RNA strand. The DNA dependent RNA polymerase binds to the promoter and catalyzes the polymerization of 5' to 3' direction on the template strand. Once it reaches the terminator sequence, the process terminates and the newly synthesized RNA strand is released. The transcription unit is a stretch. What is it? It is stretch, and the DNA transcribed into an RNA molecule. That is a frame or the open reading frame, you can say, and its function is to encode a gene, at least one gene. And suppose if the gene encodes a protein, that mRNA is produced by the transcription. A protein encoded by the DNA transcription unit may comprise a coding sequence too. And compared to the DNA replication, the transcription has a lower copying fidelity. So here you can see it is a transcription unit given in this figure or diagram. It's a promoter, it's a template strand of the DNA, it's a structural gene. Obviously, it's a structural. This is a coding strand which is expressed, which will be expressed as a protein in later phase during the translation, and this is the terminator portion of the transcription unit. The transcription of DNA includes a promoter, as I've told you, the structural gene, and a terminator. The strands that has a polarity of 3' to 5' acts as a template and are called template strands, and other strand is called the coding strand. So here it's the transcription start site, it's the structural gene and template strand, the terminator, and this is the coding strand. The blue one is the template strand, from where the DNA or the RNA needs to be synthesized. Okay. The two strands of the DNA in the structural gene of a transcription is termed as the template strand and the coding strand, and the strand that has a polarity of 5' to 3' to 5' acts as a template and is referred to as template strand. The other strand, which is a polarity of 5' to 3' is referred to as the coding strand, as you can see very clearly in this picture. The promoter and the terminator flank the structural gene in the transcription unit, and the promoter is located towards the 5' end upstream of the structural gene, which provides the binding site for the RNA polymerase enzyme. That is that enzyme which is required for the transcription. And the terminator is located towards the 3' end downstream of the coding strand, which defines the end of the process of the transcription. So promoter is the start site, terminator is the end site. Okay, here is a difference between the template strand and the coding strand. You can see it is a DNA strand with 3 to 5 prime polarity. The DNA strand with 5 to 3 prime polarity is the coding strand, as the acts as a template for transcription and codes for RNA. It does not code for any region of RNA during the transcription. So let me discuss about the mechanism or the stages of transcription. The process of transcription can be divided into 3 stages, that is initiation, elongation, or the termination and termination. Initiation of the RNA chains. The initiation of the transcription involves the 3 steps again, and the RNA polymerase holoenzyme binds at the specific promoter region in the DNA lying in the upstream of the transcription initiation site. Nucleotide sequences of the DNA preceding the initiation site of the transcription are referred to as upstream sequences. Those following the initiation site are called the downstream sequences, and the upstream bases are numbered with minus prefixes and the downstream bases are numbered with the plus prefixes. In prokaryote or the E. coli, the midpoint of the promoter sequence occur at about 10 to 35 nucleotide pairs between the transcription initiation site and the sequence respectively, and in the non-template strand and the minus 10 sequences consist the sequence, that is the Tata box is present. I'll be discussing this T, A, T, A, T, 3' end, and the 35 minus sequence is known as 5 to 3', T, T, G, A, C, S, 3'. In the eukaryotes, the two principal promoters are Tata box and the cat box, CAAT that is, and the Tata box is centered at about the position of minus 30 and has consistent sequence of 5' to 3'. In the non-template strand, the cat box is positioned near the 80 and has a consensus sequence of 5' to 3' end in the non-template strand. Now there is localized unwinding of the two strands of DNA by RNA polymerase. First, this results in the formation of the single-stranded DNA, which is free to base pair with the incoming ribonucleotides. And the ribonucleotides that form the complementary base pair with the DNA template are joined by the phosphodiester bonds and the nascent RNA is produced. So in the initiation, I was saying in the brief, I am just saying now, the transcription is catalyzed by an enzyme RNA polymerase. It attaches to and moves along the DNA molecule until it recognizes a promoter sequence, which indicates the starting point of the transcription. There may be a multiple promoter sequence in a DNA molecule, and transcription factors are proteins that control the rate of transcription. They too bind to the promoter sequences with the RNA polymerase. Once bound to the promoter sequence, the RNA polymerase unwinds the promoter of the DNA double helix, exposing the bases on each of the two DNA strands. Next, coming to the elongation. Okay. This is catalyzed by the RNA polymerase enzyme, which first unwinds the DNA, polymerizes the ribonucleotides paired on the DNA template, and lastly, after releasing the RNA strand, it rewinds the complementary DNA strands into its original structure. So, in the elongation, one strand is a template strand and is read into the 3' to 5' direction, and so it provides a template for the new mRNA molecule. And the other strand is referred to as a coding strand. This is because the base sequence of the new mRNA is identical to it, except for the replacement of the thymine bases with a uracil. The incoming ribonucleotides are used by the RNA polymerase to form the mRNA strand. It does this using complementary base pairing. I think it's very difficult and more complicated than the replication process. A to U, T to A, and C to G and G to C, the RNA polymerase then catalyzes the formation of the phosphodiester bond between the adjacent ribonucleotides. Bases can only be added to the 3' and so the strand elongates in a 5' to 3' direction. So the locally unwind segment of the DNA is known as the transcription bubble, which moves along the length of the DNA and the RNA polymerase. As the chain elongation proceeds towards the 3' to 5' end of the DNA or RNA, the nascent RNA of the 5' end is displaced from the DNA template and the RNA polymerase moves along the DNA molecule. The sequence of the events of unwinding the DNA base pairing of the ribonucleotides and their polymerization and rewinding the DNA continues until a termination signal is appeared as a 3' going end of the RNA. At last, the transcriptional termination, the elongation will continue until the RNA polymerase encounters a stop sequence. At this process, the transcription stops and the RNA polymerase releases the DNA strand, as you can see. The termination of the RNA chain occurs when the RNA polymerase encounters a termination signal or the stop signal. When this occurs, the transcription complex dissociates and releasing the nascent RNA molecule in prokaryotes. The termination signal may be produced by Rho protein or the hairpin loop. In eukaryotes, the endonucleolytic cleavage of the transcript at the 3' end occurs by a polyadenylation signal is produced, which has the consistent sequence of A, U, AA and the 3' end of RNA. In the later slides, I'll be discussing about the prokaryotic transcription and the eukaryotic transcription separately. Let's move on. Here, as you can see, the promoter is located at the, what I was saying, the 5' end that binds the enzyme RNA polymerase to start the transcription. Promoter is necessary. Sigma factor is also necessary, which helps in the initiation of the transcription. The terminator is located at 3' end of the coding strand and usually defines the end of the transcription, as I've told, where the Rho factor will bind to the terminate transcription. As there was no figure, I think you had a difficulty in understanding, but here it's very much clear. You can see its promoter, it's RNA polymerase, and it's going like this, and this is DNA helix. And upon elongation, there is a transcription bubble and a stop sequence where the transcription occurs. You may refer to these pictures also. RNA polymerase, this is a sigma factor, this is DNA helix, and upon binding of this factor, the transcription started and as well as stopped when there is a Rho factor present. What the stop signal, you can say. So, what are exons? Let me introduce. Exons are those signals which appear in the mature and processed RNA. Exons are interrupted by the introns. Introns do not appear in the mature and the processed RNA. In eukaryotes, there are 3 different RNA polymerase enzymes, 1, 2, and 3. They catalyze the synthesis of all types of RNA. The RNA polymerase 1, rRNAs, RNA polymerase 2, the mRNA. The RNA polymerase 3, tRNA. The mRNA provides the template. The tRNA brings the amino acid and read the genetic code. The rRNA play a structural and catalytic role during the translation. So, I'm going to give a brief description about the messenger RNA, rRNA, and as well as the transfer RNA. In both prokaryotes and eukaryotes, 3 principal RNAs are produced. There are the messenger RNA, ribosomal RNA, and the transfer RNA or the soluble RNA. The messenger RNA, a kind of RNA molecule that is transcribed from the structural gene and that serves as a template on which a polypeptide is constructed, is known as messenger RNA. So the function of the mRNA is to provides that codon and dictates the sequence of the amino acids present in a polypeptide chain. A sequence of 3 adjacent nucleotides in mRNA molecule specifying either an amino acid or a stop signal in the protein synthesis is known as the codon. Okay? And the protein synthesis starts with the initiation codon present at the 5' end, ends with the termination codon present at the 3' end, as you can see, of the mRNA. Next, coming to the ribosomal RNA. The RNA molecules, those remain as components of the ribosomal unit and play an important role in the protein synthesis, is known as ribosomal RNA or rRNA. What are the functions of this rRNA? Ribosomal RNAs are the structural and catalytic components of the ribosomes which provide a place of working bench for the protein synthesis. The RNA present in the smaller ribosome or subunit provides a complementary base sequence with which a special sequence of mRNA molecules shine diagonal can form The base pairing and binding. The binding is essential for the formation of initiation complex during the protein synthesis. At last but not the least, the transfer RNA or the soluble RNA. What is the definition? The small RNA, which is a clover leaf-like structure having an anticodon site and an amino acid attachment site that carries the amino acid and can translate a codon of mRNA into an amino acid in protein synthesis is known as transfer RNA or the tRNA. What is the function? The tRNA carries the amino acids to the site of the protein synthesis. I'll be discussing this in detail during the translation process. That will be more clear than from now. An amino acid binds with the 3' OH and the TH and the tRNA molecule. The tRNA molecule binds with the triplet codon of the mRNA by its anticodon site by the specific complementary base pairing. And in this way, the specific tRNA carrying the specific amino acid and having the specific anticodon can translate the specific codon to the mRNA. So the primary transcript contains both exon and the intron. Exon part is which codes for a protein, and the intron does not code for a protein, or that is non-functional. It undergoes the process of the splicing in which the introns are removed and exons are joined in a defined order. The hnRNA, the heterogeneous nuclear RNA, undergo additional processing code known as capping and tailing. In this capping in as unusual nucleotide, the methyl guanosine triphosphate to the 5' end of the hnRNA is done. In the tailing, polyadenylate tail is added at a 3' end in the template as an independent manner. Here is a clear view of the difference between DNA replication and RNA transcription. Here, the two new molecules of a double-stranded DNA are formed. One new molecule of the single RNA strand is produced. Adenine on one strand binds to the thymine, a new strand being created. Adenine on the DNA binds to the uracil on the new strand being created. The entire chromosome is replicated. Only a small portion of the DNA molecule is transcribed to the RNA, and this varies based on the cell's needs at the time. And the enzymes required the DNA polymerase enzymes are RNA polymerase. It occurs in the nucleus. It also occurs in the nucleus. Here, the process of transcription in the prokaryotes. Let's have a look, or at a glance, or the summary you can say. Its initiation, as I've described, the RNA polymerase binds to the promoter and initiates the transcription. Initiation factor, or the sigma, recognizes the promoter of the DNA and the elongation. The RNA polymerase facilitates the opening of the helix and continues the elongation. The RNA polymerase uses the nucleoside triphosphate as a substrate and polymerization in a template-dependent fashion following the rule of the complementarity. And a short stretch of the RNA remains bound to the enzyme. In the termination part, once the polymerase reaches the terminator region, you can see of the RNA polymerase binds with the termination factor Rho to terminate the transcription. Its end here. And the nascent RNA falls off with the RNA polymerase, which results in the termination of the transcription. The transcription or the translation can be coupled in a bacteria, as the mRNA does not require any processing to become active, and so the transcription and translation takes place in the same compartment. I hope you understood, because I explained this process twice in this video. Now, the transcription process in the eukaryotes I'll be discussing, and there are two additional complexities in the eukaryotes. And the first complexity is that there are at least 3 RNA polymerases in the nucleus, and the RNA polymerase 1 transcribed 28S, 18S, and 5.8S, the ribosomes. The RNA polymerase 3 is responsible for the transcription of the tRNA, rRNA, and snRNAs. You can see the RNA polymerase 2 transcribes the precursor of the mRNA and the heterogeneous nuclear RNA, which I have told in the previous slides. The second complexity is that the primary transcripts contain both the exons and introns and are non-functional. Introns are non-functional, so it needs to be removed or spliced out. The primary transcripts are subjected to a process called the splicing, where the introns are removed and the exons are joined in a defined order to become it functional, to make it functional, to code for a protein, to give a matter which could work for our body to become functional. And RNA Asian RNA undergo two additional processing called the capping as well as telling, which I've also explained. In the capping, an unusual nucleotide, the methyl guanosine triphosphate, is added to the 5' end of the HRNA, as you can see here. In telling, the adenylate residues 200 to 300 are added at the 3' end in a template-independent manner, and the fully processed Asian RNA is called the mRNA, and mRNA is transported out of the nucleus for the translation into the cytosol. Okay, and this is in this figure you can clearly see that the mRNA, its intron and exon, its splicing is done. After that, introns are spliced off, it's removed, and after that polyadenylation, it's a chain of phosphate groups of A was added, okay, to protect. And capping is also done. Capping 5-methylguanosine cap, methylguanosine triphosphate cap in the end of the hnRNA, which is known as the pre-mRNA called, and then after that matured messenger RNA or RNA is mRNA is formed, which is gold, and translate into a protein in the translation portion. So it's the process occurs in the eukaryotes. I hope I described very clearly and made easy because I explained twice in this video. So I hope you liked this video and may refer to your friends also. And RNA processing, I'll be not discussing about this because this side is additional. For your reference only, the capping, polyadenylation and splicing in details. Okay, the earlier process which I described, the eukaryotic transcriptional process, the methylated guanosine cap is added to protect the mRNA function. Addition of the methylated guanine, it offers 5' of the mRNA and protects the mRNA from degradation. The polyadenylation, it also protects the mRNA from degradation, and the endonucleases cleave the mRNA specific sequence, and the enzyme poly-A facilitates the addition of the several adenine nucleotides. Okay, and splicing, non-coding sequences, that is introns are removed by spliceosome excision process, and the coding sequences of the exon joined together by ligation by the DNA ligase, and thus the several process can be made from a single pre-mRNA. A mature mRNA is obtained at the end of the transcription. Thanks for watching. Please share, like and subscribe, and also comment in below so that I can improve much over it. Hope you like it. So have a great day.